Welcome to Microbiology classroom. Today I want to discuss about the microbe plant associations, microbe plant interactions, Rhizosphere and Phyllosphere. Microbial plant interactions, Rhizosphere and Phyllosphere. What is Rhizosphere? The Rhizosphere refers to the zone of soil surrounding the roots of plants where more microbial activities occur than roots free soil. Rhizosphere, this is the area surrounding the roots of the plant. Surrounding the roots of the plant, there is more microbial activity compared to root free soil. This area, zone surrounding the roots of the plant is called Rhizosphere. So the area of the soil, the region of the soil surrounding the plant root is more in number of microorganisms than root free soil. This is because roots release several nutrients, plant roots release several nutrients into the soil. By absorbing these nutrients, microorganisms readily grow in the zone, in the area surrounding the roots. So there is more microbial population, more microbial activity surrounding the roots of the plant than the root free zone. So this region is called Rhizosphere. Rhizosphere soil, the Rhizosphere soil is the thin layer of soil attaching to the root system. Surrounding the roots, the layer of the soil, the thin layer of the soil surrounding the root system is called Rhizosphere. Rhizosphere soil is the thin layer of soil attached to the root system. Actually, the term Rhizosphere was coined in 1904 by the German scientist Lorenz Hiltner. Lorenz Hiltner first used the term Rhizosphere. Rhizosphere was first used in 1904 by German scientist Lorenz Hiltner. Rhizo means root, sphere means the area around the root. So Rhizosphere, it refers to the surrounding zone of the root. Additionally, Rhizosphere can be defined as the region extending a few millimeters from the surface of each root where microbial population of the soil is influenced by the chemical activities of the plant, biochemical activities of the plant. So Rhizosphere can be defined as the region of the soil extending a few millimeters from the surface of each root where the microbial population of the soil is influenced by the biochemical activities of the plant. So plant produces number of metabolites and secretes these number of compounds, amino acids, growth promoting substances and several other organic compounds, vitamins into the soil. Using these compounds, microorganisms rapidly grow, multiply. So the area surrounding the root, there is more microbial activity, more number of microorganisms compared to the root free soil. So this area surrounding the root is called Rhizosphere. There is another term, rhizoplane. What is rhizoplane and what is Rhizosphere? Rhizosphere means the area of the soil, few millimeters of the soil zone surrounding the roots. Whereas the rhizoplane means the soil particles attached to the roots, the plant root surface usually including the adhering soil particles is called rhizoplane. The plant root surface including the adhering soil particles is called rhizoplane. Rhizosphere means the area of the soil, immediately surrounding the roots is called the Rhizosphere. Compared to rhizosphere area is more. Rhizosphere, it includes a few millimeters soil layer from the roots, whereas rhizoplane is the surface of the root and adhering to the soil particles. What is rhizosphere effect? Rhizosphere effect. Rhizosphere effect, plant roots release number of nutrients. Number of nutrients such as amino acids, vitamins, growth factors, other organic compounds, carbohydrates, and several secondary metabolites are released by the plant into the soil. So using these nutrients released from the roots, microorganisms will grow rapidly. Nutrients released from roots supports the rapid growth of microorganisms and survival of the microorganisms in the rhizosphere area. The roots of the plants release several compounds, several organic nutrients which favor the growth of certain nutritional types of bacteria. This influence is known as rhizosphere effect. The nutrients, organic nutrients released by the roots favor the growth of a particular nutritional types of bacteria. As a result, these nutritional group of bacteria grow rapidly and microbial number increases in the rhizosphere. So this effect is known as rhizosphere effect. Microbial populations in rhizosphere provides help to the plant in providing nutrients, and they inhibit the growth of several phytopathogens, several plant pathogens. So microbial population in rhizosphere, they provide the help to the plant by making availability of different nutrients. They solubilize the phosphate and they convert the non-available form of the rock phosphate to available form of the orthophosphate, and they solubilize the phosphate and they release the siderophores which bind to the iron and making it available to the plant, and they inhibit the growth of plant pathogens, antagonistic effect against several phytopathogens. In that way they protect the plant from the attack of phytopathogens. And as a result of this interaction, there is a qualitative and quantitative changes in the surrounding environment in the rhizosphere area. So what is the quantitative effect? The quantitative effect is the major effect observed. The number of microorganisms increases surrounding the root. So in the rhizosphere area, there is more number of bacteria, more number of fungi, more number of actinomycetes compared to the non-rhizosphere area. The number of bacteria, fungi and actinomycetes in the rhizosphere area exceeds 10 to several hundred times compared to non-rhizosphere soil. So the number of microorganisms in soil surrounding the roots is 10 to several hundred times more compared to the number of microorganisms in root-free soil. This ratio is called R by S ratio, rhizosphere effect. This rhizosphere effect quantitatively calculated by calculating the R by S ratio. What is this R by S ratio? R by S ratio is calculated by dividing the number of microorganisms in the rhizosphere soil by the number of microorganisms in the root-free soil. So number of microorganisms in rhizosphere soil divided by number of microorganisms in non-rhizosphere soil. So this ratio gives the quantitative effect of the rhizosphere. So rhizosphere effect is equal to R by S, number of microorganisms in rhizosphere soil divided by number of microorganisms in non-rhizosphere soil. R by S ratio, rhizosphere effect ratio, R by S ratio generally ranges from 5 to 20 and up to 100. So generally the rhizosphere effect R by S ratio generally ranges from 5 to 10, 20 and up to 100. Greater rhizosphere effect is seen in case of bacteria. So than the actinomycetes, than the fungi. So there is more increase in the bacterial number in the rhizosphere compared to non-rhizosphere soils. So greater rhizosphere effect is seen in case of bacteria. Generally R by S value for bacteria is from 10 to 100. It is more compared to actinomycetes and fungi. The number of algae and protozoa do not much differ in rhizosphere and root-free soil. Non-rhizosphere, if you compare how many algae are present in rhizosphere and how many algae are present in non-rhizosphere soil, there is not much difference. In case of protozoa also, there is not much difference in the number of protozoans in rhizosphere soil and non-rhizosphere soil. But if you see, the greater rhizosphere effect is seen in case of bacteria. So R by S ratio for bacteria is generally 10 to 100. 10 or 20 or more than that up to 100. So for actinomycetes and fungi also rhizosphere effect is seen. The ratios will be around the 10 or 20, but the greater rhizosphere effect is seen in case of bacteria compared to actinomycetes and fungi. Actually, the viable count are generally performed to enumerate the number of microorganisms in rhizosphere and non-rhizosphere soils. Viable count, colony count is performed by performing the serial dilution of the rhizosphere soil and non-rhizosphere soil and plating these dilutions on the medium, and after incubation colony count is performed and the number of microorganisms present in 1 gram soil is estimated by using this technique. Number of bacteria present in rhizosphere soil and non-rhizosphere soil per 1 gram is calculated. This is the table showing the number of bacteria, number of actinomycetes and number of fungi in the non-rhizosphere and rhizosphere soils of Dolichos lab. So you can see the difference clearly in case of the bacteria. So if you see the bacterial number on first day, you can see it is almost the same in rhizosphere and non-rhizosphere area after plantation, but after 10 days, 15 days, you can see the count rhizosphere effect. You can see the 20 days there are several fold increase, hundreds of fold increase in the number of microorganisms. So you can see for 15 days and 20 days how many number of bacteria are there in the rhizosphere soil. Similarly, in case of actinomycetes also, on first day there is almost same in rhizosphere and non-rhizosphere soil number of bacteria, but after Dolichos lab plantation, after 20 days, 15 days, 20 days, you can see how many folds the number of rhizosphere bacteria are increased. So it is around 95. It is around 95. The R by S ratio is 83, 95 for 15 and 20 days. You can see. In case of fungi also, at first day of the plant, on first day it is almost similar, but after 10 days or after 15 days or after 20 days, there is rapid increase in the number of fungi, at least 10 times, 20 times increase. R by S ratio is 10 to 20 for fungi. For actinomycetes, it is also 10 to 80, but for particularly bacteria, it is hundreds. So R by S ratio is in hundreds. The factors influencing the rhizosphere effect. There are several factors influencing the rhizosphere effect: soil type, moisture, pH, temperature, age and conditions of the plant. These are the several factors influencing the rhizosphere effect. The rhizosphere effect increases with the age of the plant and reach maximum at the stage of greater vegetative growth. So the rhizosphere effect increases with the age of the plant and it reaches the highest peak stage when the greater vegetative growth of the plant. Following the death of the plant, the microbial population reverts gradually to the level of the microbes in the non-rhizosphere soil. There are different compounds, different organic compounds are present in the root exudate which promote the growth of the microorganisms in rhizosphere soil. Various organic compounds present in the root exudates include amino acids, sugars, fatty acids and sterols, vitamins, organic acids, nucleotides, flavonoids and enzymes and several other miscellaneous compounds. So amino acids such as aspargine, alanine, glutamine, aspartic acid, leucine, valine, serine, glycine, cysteine, cysteine, methionine, phenylalanine, tyrosine, threonine, these are found in the root exudates. Several sugars like glucose, fructose, sucrose, maltose, galactose, rhamnose, ribose, xylose, arabinose, raffinose are released from the root. Fatty acids, palmitic acid, stearic acid, oleic acid, linoleic acid, linolenic acid, cholesterol and campestyrol and stigmasterol, cytosterol, these are reported in the root exudates. Vitamins like biotin, thiamine, niacin, pantothenate, choline, inositol, pyridoxine, para-aminobenzoic acid, N-methyl nicotinic acid are reported in the root exudates. Organic acids such as tartaric acid, oxalic acid, malic acid, acetic acid, propionic acid, butyric acid, succinic acid, fumaric acid, glycolic acid, valeric acid, malonic acid are reported in the root exudates. Nucleotides and flavonoids nucleotides like adenine, guanine, uridine, cytidine and flavonoids and enzymes like invertase, phosphatase are found in the root exudate. Several other compounds like auxins and hydrocyanic acid, glycosides, saponins, organophosphorus compounds, growth inhibitors, growth stimulants are found in the root exudate of plants. These root exudates, they provide best conditions for the increased occurrence and rapid growth of microbial flora. Algae, fungi and bacteria, actinomycetes, particularly bacteria, actinomycetes, fungi, they grow using these root exudates. So rhizosphere effect on algae and protozoa is negligible, but there is more effect of rhizosphere on the population of bacteria and actinomycetes and fungi. So these rich food materials favor the growth of bacteria, actinomycetes and fungi in the soil. So these organisms, particularly utilizing these nutrients, they grow rapidly and their population increases. The substances released by the plant roots into the surrounding soil include several things, that is amino acids, sugars, vitamins, organic acid, nucleotides, flavones, enzymes, fatty acids, sterol, hormones like phytohormones like auxins, gibberellins, glycosides, organophosphorus compounds, etc. Which microorganisms predominate in the rhizosphere? So rhizosphere microorganisms, in rhizosphere gram-negative rod-shaped bacteria are dominant compared to other group of microorganisms, other group of bacteria. In rhizosphere gram-negative bacilli, gram-negative rod-shaped bacteria are predominant compared to gram-positive bacilli and cocci. So gram-positive bacilli and gram-positive cocci are less in number. In rhizosphere, gram-negative rods are predominant, gram-negative bacilli are predominant. So these gram-negative bacilli include Pseudomonas, Achromobacter, Azotobacter, Azospirillum, Acetobacter, nitrogen-fixing bacteria, phosphate solubilizing bacteria. These are found in the rhizoplane and rhizosphere. So dominant organisms in rhizosphere soils are Pseudomonas and Achromobacter. These are the most common bacteria found in the rhizosphere and they colonize very aggressively in rhizosphere. Pseudomonas species and Achromobacter species are most common bacteria found in the rhizosphere. They colonize very aggressively in rhizosphere. These are called plant growth promoting rhizobacteria. Actually, these species, they promote the growth of the plant, so they are called plant growth promoting rhizobacteria. There are several bacteria such as Pseudomonas, Achromobacter, Azotobacter, Azospirillum, Acetobacter, and several nitrogen fixing bacteria, phosphate solubilizing bacteria are found in the rhizosphere and rhizoplane. So nitrogen fixation, some microorganisms such as Azotobacter, Azospirillum, these organisms which are predominate in the rhizosphere, they fix atmospheric nitrogen. They convert atmospheric nitrogen into ammonia and making available to the plant. So this type of nitrogen fixation is called associative nitrogen fixation. Azotobacter, Azospirillum, they are present in the surface of the root, soil attached to the roots, and they are associated with the plant roots, and they fix atmospheric nitrogen and they convert atmospheric nitrogen into ammonia, so that ammonia is available both to the bacteria and the plant. This type of nitrogen fixation is called associative nitrogen fixation. So Azotobacter and Azospirillum, they are associated with the rhizome root and they are responsible for nitrogen fixation. Some fungi inhabit the root surface, rhizoplane and rhizosphere in mycelial state. So they belong to the genera Mortariella, Cephalosporium, Trichoderma, Penicillium, Gliocladium, Gliomastix, Fusarium, Cylindrocarpon, Botrytis, Mucor, Poma, Pythium and Aspergillus. Exudates released from the roots, they cause following effects on soil microbial flora. Root exudates influence the survival and multiplication of root infecting pathogens. So the root exudates which are released from the root, they using these compounds, even root infecting phytopathogenic microorganisms, they survive and they multiply using the nutrients. So phytopathogenic population also increases due to rhizosphere effect. Root exudates stimulate the germination of the spores or sclerotia of many pathogenic fungi such as Rhizoctonia, Fusarium, Sclerotium, Phytophthora, Pythium, Colitotrichum, etc. In that way they are harmful. But root exudates, they provide nutrients for growth of even antagonistic microorganisms such as Streptomyces and Trichoderma, which suppress the growth of plant pathogenic microorganisms. So this is a beneficial effect of the rhizosphere. Root exudates, they stimulate the growth of antagonistic microorganisms such as Streptomyces and Trichoderma, which are antagonistic. They inhibit the growth of plant pathogenic microorganisms. In that way they protect the plants. Root exudates contain certain toxic substances such as glycosides and hydrocyanic acid HCN, which inhibit the growth of plant pathogens, which inhibit the phytopathogenic microorganisms and protect the plants. Root exudates play key role in neutralizing the pH of the soil and they alter the microclimate of the rhizosphere by releasing the water and CO2. Influence of rhizosphere microorganisms on the plant. Rhizosphere microorganisms, they cause several effects. They are involved in several effects. Rhizosphere microorganisms, they convert organic compounds into inorganic compounds that are absorbed by the plants. They play a major role in mineralization, conversion of complex organic compounds into simple minerals. This mineralization process was carried by the rhizosphere microorganisms so that these minerals, nutrients are available to the plant. So this is one important effect of the rhizosphere microorganisms in converting the complex organic compounds into simple compounds which the plants can absorb. So this process is called mineralization. Rhizosphere microorganisms increase the availability of phosphate in the soil by converting the unavailable form of the phosphate, rock phosphate, into apatite rock phosphate into soluble form of the phosphate, orthophosphate. So rhizosphere microorganisms solubilize the rock phosphate they convert non-available form of the rock phosphate to available form of the phosphate, orthophosphate to the plants. In that way, they solubilize the phosphate, making it available to the plants. Rhizosphere microorganisms, they increase the uptake rate of calcium by the roots. Rhizosphere microorganisms, they also produce plant growth promoting substances such as auxins and gibberellins, which promote the seed germination and growth of the plant, development of root hairs and plant growth. So rhizosphere microorganisms, in that way, they promote the plant growth by producing the phytohormones like auxins and gibberellins. Rhizosphere microorganisms also synthesizes vitamins, growth regulators. These vitamins and growth regulators released by the rhizosphere microorganisms promote the plant growth. Here microorganisms such as Azotobacter, Azospirillum, Achromobacter, Enterobacter, Klebsiella, Bijerinkia, Clostridium, Erwinia, Derxia, Frankia, Pseudomonas, etc. fix atmospheric nitrogen, gaseous nitrogen into ammonia. So this ammonia used by the plant, this type of nitrogen fixation is called associative nitrogen fixation. Rhizospheric nitrogen fixation in crop field is estimated as 40 kg of nitrogen per hectare per year. So some rhizosphere microorganisms synthesize vitamins, amino acids, growth regulators that stimulate the plant growth. Some rhizosphere microorganisms, particularly belonging to the genera of actinomycetes such as Streptomyces, Actinomyces, etc., produce antibiotics which inhibit the growth of plant pathogens. Some rhizosphere organisms exhibit antagonistic effect against plant pathogens, trichoderma, fungi and pseudomonas bacteria. So these organisms, they inhibit the growth of plant pathogens and suppress the growth of plant pathogens. Secretions of microorganisms help to combine soil particles into soil aggregates around the plant root. These aggregates hold the moisture. So in that way, they are helpful in maintaining the structure of soil. Phyllosphere. What is phyllosphere? The area surrounding the leaf is called phyllosphere. Stems, leaves, fruits of plants provide suitable habitats for microbial populations. So epiphytic microorganisms, they grow on the surface of stems, leaves, fruits of the plants. The habitat adjacent to the plant leaf surface is known as phyllosphere. The habitat adjacent to the plant leaf surface is known as phyllosphere. And habitat directly on the surface of the leaf is called phylloplane. The habitat adjacent to the plant leaf surface is known as phyllosphere, and the habitat directly on the surface of leaf is called phyllosphere. Phyllosphere refers to the zone on leaves inhabited by microorganisms, whereas the phylloplane is the surface of the plant. Phylloplane, it represents the plant surface, plant leaf surface. Phylloplane represents the plant leaf surface, whereas phyllosphere refers to the zone of leaves inhabited by the microorganisms. Phyllosphere is the immediate area of the leaf, and whereas phylloplane is the actual leaf surface. So phylloplane is the leaf surface, whereas the immediate area of the leaf is called phyllosphere. Floroplane is the term used for the microbial community associated with flowers. The term phyllosphere was coined by Dutch microbiologist Runen. Runen, he coined the term phyllosphere after her observations on Indonesian forest vegetation, where thick microbial epiphytic associations exist on leaves. After a study on Indonesian forest vegetation, where observed the microbial association on the leaves, Dutch microbiologist Runen coined the term phyllosphere. Leaves are exposed to dust and air currents. Dust and air continuously fall on the dust falls on the surface of the leaves and air currents continuously strike the leaves. So dust and air currents resulting in the establishment of different microbial flora, typical microbial flora on the surface of the leaves. Of course, the leaves contain cuticle, wax and other appendages. Cuticle, wax and other appendages of the leaves help in attachment of the microorganisms to the leaves. In tropical rainforest, the Phyllosphere bacteria produce gums or slimes which presumably help bacterial cells to attach to the leaves under the effect of heavy tropical rains. Microorganisms may die, survive, proliferate on leaf depending on the extent of leaf exudates. Microorganisms may die or survive or multiply, proliferate on the leaf depending on the leaf exudates or diffusates. The leaf exudates include amino acids, sugars like glucose, fructose, sucrose, etc. Under high humidity in rainforest of tropical and temperate zones, the microflora of leaves may be very high. So in high humid conditions, the microorganisms number is very high compared to low humid conditions. So the Phyllosphere is the region of the plant leaves. The Phyllosphere is the aerial region of the plant leaves colonized by the microorganisms. So there are different types of microorganisms are present in Phyllosphere. Bacteria, fungi, algae, protozoa and nematodes inhabit the leaf and stem surfaces. But the most abundant microorganisms in Phyllosphere are bacteria. Bacterial number may average 10 power 6 to 10 power 7 bacterial cells per centimeter square. So 1 square centimeter area contains on leaves, 1 square centimeter area on leaves contain 10 power 6 to 10 power 7 bacteria. The most dominant group of microorganisms in Phyllosphere are bacteria. Their population may be 10 power 4 to 10 power 5 per millimeter square of leaf surface and up to 10 power 8 bacteria per 1 gram of leaf material. With that, you can assume how many bacteria are associated with the Phyllosphere. Some famous eukaryotic microorganisms associated with the Phyllosphere are yeast, wild and indigenous yeast, Cloquera, Metchanicovia, Pichia. These species are associated with Phyllosphere. For example, grapes, they contain very high number of yeast. Grapes contain very number of these yeast such as Cloquera, Metchanicovia and Cloquera, Metchanicovia and Pichia than Saccharomyces. Saccharomyces, number of Saccharomyces are very less compared to these yeast, Metchanicovia, Cloquera and Pichia. Of course, Saccharomyces is also found, but its number is less in Phyllosphere of the grapes. These wild yeast are thought to impart spoiled taste to the wine when used in the fermentation. That's why after making the pasteurization of the grape juice, Saccharomyces cerevisiae is introduced for making the wine. Actually, the Phyllosphere is a hostile environment due to rapid changes in temperature, humidity and limiting of the nutrients and solar irradiation. So solar irradiation, it contains UV light. Solar radiation, it has UV, it damages the microorganisms. However, microorganisms adapt to these conditions. Phyllosphere microorganisms, they adapt to cope these conditions. Many bacteria are pigmented to prevent UV damage. So they have some pigments to prevent the ultraviolet light damage. So microbial communities, they develop along veins and around trichomes and stomata where nutrients leakage from the plant surface. Particularly in Phyllosphere area, microbial community preferentially develop along the veins on leaf and around the trichomes on the leaf and stomata. So there the microbial number is more compared to other areas on leaf. Moisture availability is the key limitation to the microbial growth on the leaf surface. Actually, the cuticle of the leaf prevents the moisture from leaving the inside of the leaf and limits the water remains on the leaf surface. To overcome this challenge, bacteria aggregate and form biofilms producing extracellular polymeric substances EPS which can resist desiccation. Some other bacteria they produce surfactants to increase the wettability of the leaf and lessen the ability of the cuticle to limit water accumulation. The leaf surface has a boundary layer with a microclimate that typically has a higher humidity than the broader Phyllosphere, and this likely mitigates some of the desiccation pressure for microbiota that are in that layer. Microbial populations typically increase following precipitation, rain, when there can be substantial changes in the diversity and composition of the Phyllosphere microbiome. The effect of moisture from dew on the leaf community has not been explored, but it is assumed that its effect is similar to rain. Rain and dew promotes the growth of microorganisms in the Phyllosphere. Phyllosphere microorganisms are allotrophs. They require low nutrient conditions, and they interact with the host plant to obtain more nutrients. Cuticular waxes are generally resistant to chemical movement. Some plant metabolites can move to the leaf surface, supporting to the microbial growth. These compounds may arrive on the leaf surface by excretion from the leaf cells or due to osmotic pressure when the leaf is wet. Plants also release volatile organic compounds which support specific populations of microorganisms. For example, methylotropic bacteria that metabolize plant-derived single carbon compounds are abundant in the abundant constituents of the Phyllosphere of many plant species. Alpha proteobacteria are particularly abundant on the Phyllosphere compared to other bacteria. Gamma proteobacteria also commonly reported in the Phyllosphere. So proteobacteria, they are metabolically diverse, and they carry out methylotropy, nitrification, nitrogen fixation, anoxygenic photosynthesis, etc. So proteobacterial population is the predominant population in Phyllosphere. Yes, bacterial and fungal populations are seen in Phylloplane and Phyllosphere. The number of microorganisms on the leaf surface depend on the season, whether it is a rainy season or it is spring season or it is winter season or it is summer season. Based on the season, the number of microorganisms on the leaf surface, type of microorganisms on the leaf surface depend on the season and the age of the leaf. Phyllosphere bacterial assemblages are generally less species rich than the Rhizosphere or soil. So Rhizosphere contain different variety of microbial species than the Phyllosphere. Phyllosphere bacterial assemblages are generally less species rich compared to Rhizosphere. Under conditions of high humidity in wet forest, in tropical and temperate zones, tropical and temperate wet forest, the microbial flora are very rich in Phyllosphere. The dominant microorganisms are Phyllosphere, just I told to you, that is proteobacteria, alpha proteobacteria and gamma proteobacteria. These are dominant microorganisms of Phyllosphere. Azotobacter, Byzantinia, Pseudomonas, Pseudobacterium, Phytomonas, Erwinia, Sarcina. These are the dominant organisms in Phyllosphere. Nitrogen fixing cyanobacteria, Anabina, Calothrix, Nostoc, Cytonema, Talipothrix have also been found in Phyllosphere of moss forest. And Phyllosphere actinomycetes include Actinomyces and Streptomyces. Actinomyces and Streptomyces are also found on Phyllosphere. Only on the surface, some endophytic microorganisms of the Phyllosphere are also found. In the endophytic, inside the leaf also, epiphytic and endophytic. Phyllosphere contains two types of microorganisms, epiphytic organisms and endophytic organisms. Endophytic bacteria are also found in the leaves. So the richness of bacterial endophytes in Phyllosphere you can see in a number of plants. So bacterial endophytes are found in number of plants. They belonging to Burkholderia, Actinomycete and Actinoplenis and number of other bacteria. Found in Phyllosphere are Cladosporium, Alternaria, Cercospora, Helminthosporium, Erysife, Spherotheca, Podospora, Uncinula, Sporobolomyces, Bullera, Cryptococcus, Rhodotarula, Tarula, Tarulapsis, Rhodotarula, these are yeast, Oidium, Puccinia, Melanospora, Saccharomyces candida are yeast, Tilletiopsis, Penicillium, Cephalosporium, Fusarium, Periconia, and Rhynchosporium, Spermospora, Ariobasidium, Collitotrichum, Metarhizium, Myrothecium, Verticillium, Pithomyces, Mucar, Cunningamella, Fusarium, Aspergillus, Carvularia, Rhizopus, Trichoderma, Heterosporium, Syncephalastrum. These are the different fungi recorded in Phyllosphere. Different microbial activities in Phyllosphere. Many bacteria present in Phyllosphere, they fix atmospheric nitrogen and provide to the plant. This is a beneficial activity of the Phyllosphere microorganisms to the plant. So in that way, plants benefit from the association of microorganisms. Many bacteria present in Phyllosphere, they fix atmospheric nitrogen. Azotobacter, Azospirillum, and several other bacteria, they fix atmospheric nitrogen and provide it to the plant. Achromobacter, Pseudomonas, Cellulomonas, Bacillus, these are the species present in the Phyllosphere of pea and wheat. This Phyllosphere microflora forms a biological barrier to the establishment of plant pathogens. They prevent the entry of phytopathogens. In that way, they protect the plant. Phyllosphere microbes control effectively the spread of airborne pathogens because they induce plant to synthesize phytoalexins which destroy the plant pathogens. Some Phyllosphere microorganisms, they synthesize growth promoters such as auxins, indolestic acid, a phytohormone auxin which enhances the plant growth. Certain Phyllosphere bacteria such as Pseudomonas syringae and Erwinia herbicola, they produce a surface protein that initiate ice crystal formation resulting in frost damage to the plant. They cause damage. Certain Phyllosphere bacteria, Pseudomonas syringae and Erwinia herbicola, they produce a surface protein which initiate the ice crystal formation, so resulting in frost damage to the plant. Thank you for listening the lecture on Rhizosphere and Phyllosphere.